Hello everyone, my name is Gulati and I'm an NHMRC Early Career Fellow at the School of Dentistry UQ. Today I'll be talking about tailored bioactivity and therapy from dual micro nano titanium dental implants. The dental implants enjoy a fairly high success rate. However, in compromised conditions such as poor oral hygiene, smoking, diabetes, or osteoporosis, the long-term stability may be compromised. Dental implants is unique because it needs appropriate osseointegration at the bone and screw level, as well as effective soft tissue integration at the transmucosal region. In high risk patients, there is a high chance of implant failure, and as a result, various surface modifications has been performed on conventional dental implants. These include topographical, chemical, or biological enhancements. An ideal implant would be the one which takes care of all bioactivity and therapeutic needs directly inside the dental microenvironment. Our approach is nanoengineering. We nanoengineer the surface of titanium dental implants via a procedure called electrochemical anodization. Anodization permits controlled self-ordering of these nanostructures, which are called titanium dioxide nanopores, on the entire surface of the dental implant. Now, these nanostructures, due to their topography, which is in nanoscale, they enhance the bioactivity, whereas the empty nanotubes or nanopores can also be used to load and locally release various therapeutics. Now, we have optimized the fabrication of various nanotopographies. You can see here nanotubes, and we have also more recently able to fabricate controlled nanotopographies such as aligned nanopores, nanotemplate, and random nanopores. The dimensions of these nanostructures can be controlled, whereas the alignment is also very important because onto these alignments, we can control how the cells behave. Very recently, we are able to fabricate controlled nanotopographies on conventional dental implants. So here you see the different nanotopographies on abutment and screw. If we just do the anodization once, we have aligned nanopores. When we remove the nanopores, we have a nanotemplate. When we perform the second anodization, we have random nanopores. Now, as I mentioned, the dimensions of these nanostructures can be controlled, and so is the alignment of the nanopores. And based on that, we can control how the cells will behave. The entire technique is very scalable, cost effective, and can easily be translated into commercial implants. We get to control the chemistry and topography of the nanostructures, as well as the nanostructures are very mechanically stable. Now here we are talking about dual micro nano surfaces. We start with a rough titanium, we polish it to have a micro machine surface, and then we perform anodization at 40, 60, and 80 volts. As a result, we have titanium nanopores, which is TNP 40, TNP 60, and TNP 80. You can see that as we increase the voltage, the diameter of the nanopores is increasing. Again, we get great control over the topography and chemistry of it because we know the cells correspond to both these features. Next, we did the atomic force microscopy, and you can see here the rough titanium is randomly rough, whereas the micro titanium is more micro machine in nature. Now, these two substrates, the rough and micro titanium, represent clinically used surfaces. And when we do anodization, we see that for the nanoporous substrates, it preserves the underlying micro roughness. Now, this is very important. And in this pioneering study, we showed that we can preserve the micro roughness of the dental implants, which is very crucial to ensure initial stability. It's noteworthy that most research with this domain of anodization generally involves polished surfaces. That means you lose the micro roughness. But in this technique, we preserve the micro roughness and we superimpose nanotopography on top of it. We cultured different cells onto the surfaces of nanopores as well as controls of micro and rough titanium. For the macrophages, you can see just by tuning the diameter of the nanopores, you see the TNP60 and TNP80, we can significantly reduce the proliferation of macrophages, whereas the proliferation of human osteoblasts and gingival fibroblasts was maintained over time. Looking at the scanning electron microscopy images at day 3, you can see that the macrophages are more circular in nature and they are not super stretched. This indicates an inactive state, whereas the osteoblast and gingival fibroblast seem to be stretched well, attached well onto these surfaces. Looking at the high magnification image for the TNP80, which is the titanium nanopore at 80 volts on day 4, we see again the macrophages in a more circular-like spreading morphology indicating an inactive state, whereas the osteoblast and fibroblast tend to align parallel to the nanopores. The yellow arrows here show the nanopores underneath. Clearly, we are able to achieve mechanical stimulation of the cells. Next, we look at the soft tissue integration, which is very important at the transmucosal region of the implant because it serves as a barrier towards the ingress of pathogens. We looked at different time points starting at 1 hour because we believe it is an initial time point which dictates the further behavior of the cells. Even starting at 1 hour, we see that the cells on the nanopores synthesize more ECM, synthesize more filopodia and stress fibers. Interestingly, from day 1 onwards, we see that the cells start to align parallel to the nanopores. The yellow dotted arrow signifies the direction of the underlying nanopores, and starting at day 1, the cells on the nanopores started aligning parallel, while at day 3, the alignment was almost perfect. Looking at high magnification image even at day 1, we see that for all the nanopores, the cells are super stretched and they are aligning parallel to the nanopores. It is noteworthy that this has not been reported before on any of the nanostructures fabricated by electrochemical anodization. And it is also noteworthy that the alignment did not happen on the micromachine surface. And you can see that all the nanopores have mechanically stimulated the cells. Looking at the day 7 image, we see that the cell alignment is further strengthened. The cellular alignment is almost perfect to the underlying nanopores, and there's a lot of stress fibers indicating strong anchoring of the cells. This is clear evidence of mechanical stimulation, and we are currently exploring the different factors involved with the mechanotransduction. Next, we look at the protein gene expression corresponding to different proteins that enable wound healing, soft tissue integration, and cell to cell or cell to implant interaction. Clearly, for the nanopores, the expression has been enhanced over time. Particularly for the TNP80, the effect is more prominent. You can see the enhanced activity of type collagen, collagen type 1, collagen type 3, and also integrin beta 1, clearly indicating the surfaces, the nanoporous surfaces, would promote a profile which is more wound healing, which is what is needed for the soft tissue integration. Now, to summarize, first of all, the technique we use, anodization, is very clinically translatable. It is cheap, cost effective. So the dental implants can be modified with nanopores using this simple anodization technique. The nanopores are highly bioactive and we get great control over the topography and chemistry of it, by means of which we can control the cell functions. The nanopores can also be used to load and locally release drugs, whereas the nanopores will survive the load-bearing oral microenvironment. Using a single surface without loading or releasing any proteins or growth factors, we are able to stimulate different cells. We are able to significantly reduce the proliferation of macrophages, which translates into immunomodulation, whereas we are able to enhance the activity of osteoblasts and fibroblasts, which are also mechanically stimulated, which translates into achieving osseointegration and soft tissue integration. Thank you. Now I would like to thank also the collaborators and Professor Sasha Ivanovsky, who is the group leader, and the funding bodies. Thank you very much for your attention.